Hello everyone, this is River. See you again. And this is a follow-up of the soil sampling video. Today I'm going to demonstrate how do I extract the DDT from the soil samples and prepare for GC analysis. In addition, I will also do a brief introduction about how do I do the data analysis. So before I do the experiments, I would like to go through some of the background. So basically here shows the soil particle. We can see that the soil particle is covered by the soil organic matters, and we also know that DDT is hydrophobic. So many of the DDT molecules, they will be absorbed or adsorbed by the soil organic matters. So what we are going to do is to extract these DDT molecules from these soil organic matters by using a solvent that is more hydrophobic than these soil organic matters. And so that DDT molecules are moved from these soil organic matters into the solvent. And then we have almost all of the DDT molecules in the solvent. And this can be used for GCMS analysis. And of course, this extraction is under certain conditions, such as high temperature and high pressure, to make this process faster. So now let's do the experiment. So here is a soil sample that I sampled last time, and it was stored in the -20 freezer. But before we do the analysis, we need to remove all of the waters from this soil sample. And here is a baked soil, and we will use this baked soil to do the analysis. And the next step is to prepare samples for ENCE. And the full name of ENCE is Accelerated Solid Phase Extraction. And here is a column of the ENCE extraction. So basically, we will fill up the sample and the clean sand within this column. The clean sand is a media that can help with the extraction. So now I am going to prepare the samples, 2 blanks and 1 matrix spikes. And so the first step is to put the filter into each of the column. And then we are going to weigh 1 gram of soil samples and fill it into each column. And then we fill up with some of the sand. And so for the first one and this one, they are all sand blanks, so we can fill up it with sand only. And the next one is matrix spike. For the matrix spike, we are going to use the clean soil instead of the sample soil. And then the next step is to add surrogates and spikes. So first, let's add surrogates. We are going to add the surrogate to each of the column. And the next step is to add the matrix spike. And then we are going to fill the sand all the way up to the top of the column, and then make the column finish. And so now we have the column ready. The next step is to move these columns to the ENCE. So now let's go to the ENCE. And so this is the ENCE, which is accelerated solid phase extraction. So first we need to put the columns into this device, and then for the corresponding position, we have this ENCE vials to collect the extract. And so And this ACE will extract the DDT in each of the column and then send the extract into the vials here. So now let's run the ACE. So for each of the extraction, it will take about 45 minutes. And so let's wait for 5 hours and then go back to check our extraction. So now it is after 5 hours, and we can see that we have some of the extract in our vial. And the next step is to evaporate the solvent so that we can concentrate the DDT samples. And so this is the evaporator, and it is connected to nitrogen. So basically, we transfer our sample into the evaporator. And we can see that it has many needles, and this needles is connected to the nitrogen gas. So basically, these needles will blow up the nitrogen to accelerate the evaporation process. And also, we can fill up some of the water in this container. So the water will heat up the temperature, and the evaporation process can be accelerated. And so we are going to transfer our sample into the evaporator. It will take about 45 minutes to dry out this solvent. And so let's wait for about 45 minutes. And so now it is after 45 minutes. And we can see that this is completely dry. And we have some crystals at the bottom of this vial, and they contain the DDT. And the next step is to add 2 ml of hexane into each of the vial. And the next step is to add internal standard. After adding the internal standards, we are going to mix the solutions. And then we are going to transfer the samples into the GC vial. And because we have 2 milliliter of samples in the vial, and for each GC vial, we transfer 1 milliliter. And so now we have prepared all the samples for the GC analysis. But before we go to the GC for the analysis, we need to add more vials into the GC sequence. So for a typical GC sequence, first we have a pure solvent, and in this experiment we use hexane as a solvent. And so this is a pure hexane, and then it follows by the standard curves because we want to calibrate the instrument first. And these are the standards for the calibration curve. And then we add our 1 set of our sample into the GC sequence. And so these are our GC vials. And the hexane is for the instrumental blank. And these standards are for the calibration of the instrument. And we have 2 sand blanks for the methods, and we have triplicate of our sample, and we also have 1 matrix spikes. And then we are going to take this to the GCMS. And so this is GCMS, and we can see that the noise is very large in this room. So I'm going to put this rack into the GC. And then I'm going to use the computer to add the sequence. And now the GCMS will analyze the concentration of DDT in each of the sample, and finally we can get the result. So for each of the vial, it will take about 1 hour for the analysis. So we need to at least wait for 13 hours. So let's just wait for 24 hours and come here tomorrow. And so now we have the GC result here. And then we are going to check the QA and QC. If the QA and QC is good, then we can calculate the average of the triplicate to represent the DDT concentration of this one sampling point, like shown on the map here. And this data can be used for further analysis. By having all of the data, like shown on the map on the left, we can do the mapping of the data on the map, like shown on the right side here. And this process is called spatial interpolation. There are several ways for the spatial interpretation, such as inverse distance method, natural neighbor method, spline methods, and trend surface methods, and so on. But these methods are deterministic methods which cannot characterize the uncertainties. So kriging is usually used for spatial interpolation, and because kriging is based on the geospatial statistics. Kriging has two assumptions, and the first assumption is stationary. It means that the probability distribution of the DDT concentration are the same everywhere, like shown on the figure in the left, and the mathematical expression is on the right side. And if we assume the concentrations follows a normal distribution, it means that the mean and variance of the concentrations are constant in this region. And the second assumption is spatial correlation. It means that the concentrations are spatially correlated, and the correlation decreases with distance. And if the 2 points are far away enough, then these 2 points are independent. For example, like shown in the figure on the left side, we can see that the concentration of point A1 is more likely to be closer to the concentration at location A than the concentration of location A2, because A1 is spatially closer to A. And this relation can be expressed mathematically by correlation function and the covariance function on the right side. And these two functions are basically equivalent. Here is the expression of kriging. Basically, kriging is a linear mixed model, and in this model, the correlation matrix is described by the covariance function like shown here. And the covariance function can be fitted by our data. And so we can plug in our data, and here is the empirical covariance function. And this plot is also called variogram. And here is a spatial interpolation result by ordinary kriging. And that's it for today. Thank you very much.